Hello, students. Extra practice with Module 9. Module 9 involved the ANOVA. We have 3 questions, and I'm going to be working in R for them. So the first question on the ALL data set, consider the ANOVA on the gene with probe 109AT. Expression values. We want to use B cell patients in 5 groups: B, B1, B2, B3, and B4. First of all, we need to conduct a one-way ANOVA. Do the mean expression values of this gene differ in the different disease stages? Well, we need to use the ALL library for this data. And then I want to use just the patients in B, B1, B2, B3, and B4. I'm using expression values for the gene 109AT. Maybe before we run the ANOVA, we can look at a plot. I will use this dot plot, which is in the library lattice. On the x-axis, we see the 5 stages, and then we have the gene expression values for 109AT. The question is the mean gene expression differ among the 5 groups? In the one-way ANOVA, we know the null hypothesis is that all 5 means are equal. And the alternative is there is some difference in the mean gene expression values. Letter A. Here we see the ANOVA. You notice the p-value is 0.01082. If we use a significance level 0.05, we would reject H0. So maybe I will comment this. At the alpha equals 0.05 level, we reject H0. There is some difference in mean gene expression values in the disease stages, but the ANOVA alone, as we know, won't tell us where the difference is. Okay, letter B. From the linear model fits, find the mean gene expression value among the B3 patients. We can use this summary. So the intercept is here. It's 6.8102. We can tell by looking at it, the intercept corresponds to patients in group B. Now we are interested in mean gene expression value in B3. So we're going to take the intercept and add this minus 0.1249. And we see the answer is 6.6853. Letter C, which groups have mean gene expression that is different from the mean gene expression in group B? Group B is corresponding to our intercept here. And so we can answer this with what's printed in this coefficients table. The p-values in the last column, starting with the second row, are comparing that group to group B. Now look at them. 0.2408, 0.0742, 0.5167, and 0.6166. None of these are statistically significant. Oh, so the answer is none, based on the last column of this summary. Okay, great. Now, letter D. The thing is, we scroll up. Our ANOVA was statistically significant, but so far we know the difference does not come from group B and any of the other individual groups. We can use this pairwise.t.test to figure out where the differences are. We are told to use FDR 0.05 level. Okay. We use this pairwise.t.test. This is all of the pairwise t-tests. So what do I see significant in this table? Only right here. So the only differences are in B2 and B4. And that is what I say here. Looking back at our dot plot, here is B2, third column. Here is B4, the fifth column. These two are the only pair that have a statistically significant difference in their groupings. Letter E, we want to check the ANOVA model assumptions with diagnostic tests. Do we need to apply robust tests here? Well, we can test for normality of the residuals. We can test for homoscedasticity as well. First of all, we will look at the Shapiro-Wilk test because I don't have an error there. Shapiro-Wilk test for normality, you notice my p-value, 0.1177. We would fail to reject the null. And so it seems reasonable that the residuals are normally distributed. Now, my issue is because I need this library LM test in order to run the Bruch-Pagan test. Now I should be able to call this Bruch-Pagan test. Okay, great. Our p-value is very large, so we fail to reject the null here. It seems reasonable that our data satisfy the homoscedasticity assumption. Okay, that was a big problem. So we are finished with the first question. We will move on to number 2, clear my saved objects. Number 2, we want to apply the non-parametric Kruskal-Wallis test for every gene on the B cell ALL patients in stages B, B1, B2, B3, and B4. Well, let me bring back the library and the data and pull off the patients in these particular disease stages. I'm going to use the apply function here. I am applying to the gene expressions this Kruskal.test function and then storing all the p-values. Takes a little time because there are a lot of genes. Okay, so we are finished. Now let's adjust for multiple hypotheses using FDR, Benjamini, Hochberg. The question is, how many genes are expressed different in some of the groups? I just want to know how many in this vector p.fdr are less than 0.05, and the answer is 423. Just as a remark about adjusting for multiple hypotheses, if you want to know the length of this p-values, 12,625. That is how many statistical tests we did. That's why it's important to adjust for multiple hypotheses. Moreover, if we did not adjust the initial set of p-values less than 0.05, we get a very large number, 2,241. But a lot of these will be false positives because we have so many hypotheses tested here. So I want to go back to our final answer, which is right here, 423. Okay, letter B, find the probe names for genes with the smallest 5 p-values. Well, if we just sort the p-values, let's see what we get. In a way, you see, we could read the probe names off of this. There are the 5. We can also call this names function, and we get them right here. So here is our answer to number 2B. You might recognize this probe name, 1866 GAT. We had that in our Canvas course and some examples. Okay, number 3. On the ALL data set, we consider the ANOVA on the gene with probe 38555AT. Expression values on 2 factors. So first of all, let me reload this library and data. First factor is disease stages. I'm going to use B1, B2, B3, and B4. Now I have loaded those patients into this all B, 1, 2, 3, 4. Okay, we have the expression values for the gene of interest. Now, as I mentioned, first factor, we have B1, B2, B3, B4. The second factor is the gender. So let me store my two factors into variables. We have stage and we have sex for gender. Okay, one thing that we did when we did a two-way ANOVA is we looked at a box plot. So let's do that here. We have 8 possibilities here where the group membership depends on both factors. We have 4 disease stages, and then this data set has 2 possible genders, male and female. Okay, so let's go back where we can run the two-way ANOVA. You notice we have a question about the interaction. So I will run the full two-way ANOVA with the interaction term. Looking at this ANOVA table, we see 3 p-values, and with a two-way ANOVA in general, we have 3 null hypotheses. So the first one, looking at this row for stage, the null hypothesis would be that there is no difference in the means of disease stage. This is a very small p-value, and so we reject that null hypothesis. It appears there is a difference in the means of disease stage. However, if you look at the p-value for gender, if you look at the p-value for the interaction, these are both very large. So we would fail to reject the null hypothesis that there is no difference in the means of gender. We would fail to reject the null hypothesis that there is no interaction. So let me just type all of this. Okay. Last part of the last question, check the ANOVA model assumptions with diagnostic tests. Are any of the assumptions violated? Well, in number 1, we were in great shape. None of the assumptions were violated. So let's check this here. If we run the Shapiro-Wilk test for normality of the residuals, well, at the alpha is 0.05 level, we would reject the null. So it does not seem that the residuals are normally distributed. We could also check the other hypothesis, which is the homoscedasticity of the residuals. Here, this looks okay. We fail to reject the null. Okay, this is the end of this extra practice worksheet. Thank you so much, students.